But there are also plenty of other tools that people have created that are still useful, maybe a little bit more niche or stuff like that. But these are still really powerful tools that can be really useful in a variety of different analyses. So the first of which, I only have 4 on here, and there's 4 collab notebooks associated with them, but this is by no means exhaustive. I want to also emphasize that. The first of which I want to talk about is ThermoMPNN, which we've talked a lot about protein MPNN. This really just builds off of that. So we have pretty much the same setup in protein MPNN, the encoder, the decoder. It takes in the backbone positions. It learns to, based on the distances between each pair of atoms in the backbones. We have those structure features. We throw that through the encoder. In the decoder, we start decoding that sequence and start making some predictions. When we use this for ThermoMPNN, the goal is quite different. Here we're trying to predict what the change in stability is for a given point mutation. So you may be asking yourself, like, why use a protein MPNN model for this? Does anybody have like any initial like feelings about that? Why use protein MPNN as a base for this type of model?
Yeah. Local context is really important for stability.
Okay. And how does like that's very true. How does the sequence design part, the things that protein MPNN has been learning about, how does that relate to stability? Does it?
Maybe I think you can you can learn the amino acid frequency or the distribution on a different part of the enzyme, right? Like this hydrophobic core and the head-feeding surface area.
Yeah, that's a good point. So it starts to learn based on the local environment of every residue, kind of what's tolerable in that environment. And so this is really useful when you're starting to make a prediction about changing that environment. So here Henry took protein MPNN and we're going to fine tune it. So based on some of the features that protein MPNN has learned, it's still going to be able to extract information about the backbone, still going to be using the information that it's learned about the local context around each residue and how that kind of prefers certain amino acid types. When we're doing transfer learning, we kind of fix a first part of a model. It's kind of pre-trained. You fix it usually, and then you add maybe a layer to an adapter at the very end to adapt the knowledge. You transfer the knowledge from the pre-trained portion into some other new task. Ideally, that task is related because you want it to require less training. It already has some sort of idea of kind of some of the properties of that task. So for example, sequence design is quite, as we've mentioned, it's similar to stability. And I think another argument as to why is that native proteins are stable, right? They're marginally stable. They might not be optimized for the most stability because they have other constraints that are governing their evolution, their functional properties, who they interact with, and stuff like that. So I think that's also maybe another argument why the sequence design model has maybe some of the stability information already present within it. So how we actually do this is we took 3 of the embedding layers from the decoder. So the decoder had 3 steps. Within it, there's just message passing neural network layers. We took the embeddings from each of those, concatenate them, and then we also tell it what sequence is the native sequence. This allows the model to think about what the native context of that residue is and how it'll start to put together how that would change if you mutate that central residue. So we take all those pieces and then we start applying some light attention. This kind of just rescales the features and the embeddings to make it more useful for the stability prediction task. And then we compare the mutant embeddings, or the native embeddings, with all 20 mutant embeddings. So we know that in this case we have a bunch of data that comes from a giant Rocklin megascale data set for stability based on this protease assay. But we have a ton of data, and it's a site saturation mutagenesis, so we literally have every position mutated to every other position for most of these proteins. Some of the data is not as nice, but we throw those away. But it's mostly complete. So we have the native sequence, and we also have the site saturation mutagenesis for all the different amino acid types at that position. We compare an embedding of that structure with that native sequence to that structure with the mutant sequence. So we don't really do much. You don't have to model the mutant structure, which is convenient because a lot of other these delta G prediction networks also require modeling of a mutant structure, which in itself constrains the problem and adds a lot of assumptions because you're making assumptions about, it's not going to blow up the protein, it's going to have the same exact shape, etc. So we take an embedding that uses the native sequence, an embedding that takes the mutant sequence, and compare them. So this kind of just makes sure that they align, and then you subtract those embeddings, really. And that will give you kind of this delta G prediction for every position, for every amino acid type. And so you can kind of get an understanding of what the model would think a favorable mutation is, as well as a destabilizing mutation, as well as maybe something that's just kind of tolerable. I know that was a lot. Are there any questions? I'd love to clarify.
Was this ever trained with any delta G data?
Yes, the Rockland dataset does have delta G data measurements.
So I think the problem is in the mega scale data set, if they use the demutation screening of specific protein and incorporate that into the mega scale data set, or they just verify different sequences.
They had a bunch of proteins, around 300 or so, but they're all mini proteins. And a lot of them were de novo, and they performed this protease activity. So they I think they linked it to the transcript of that protein, and then depending on the stability of that protein, they can correlate the amount of cleavage and how much sequence they could read from the sample still, and they can correlate that with delta G. Yeah.
I can understand that, but I think what I want to ask is about their library. Is the mutational library, or
Yes.
Yeah, they do deep mutational screening on each protein.
Yes, it's completely site-saturated mutagenesis. So every position is mutated to every other position, and yeah, they try to measure the stability for those mutants. Yeah, Ben.
Do you think it's important to include the embeddings of that mutation in each decoder layer, or from each decoder layer, or do you know why he took?
So yeah, so these are really kind of taking into account the native structure of that protein. So it's learning from what the native sequence is, as well as the structural information of that protein. So it has an idea of why this position is the way it is, and kind of has chemical properties and maybe some physical properties of that as well. And I think that's kind of how it starts to learn to if you change that, how it affects things. The mutant sequence doesn't come in really until the end. It's a little hard to see from this diagram, but yeah. Yep. Yeah.
So could you elaborate a little bit on the dataset?
The dataset is this mega scale Rockland dataset. So it's the biggest stability dataset that we've seen in ever since then. And it's I guess I kind of went over it a lot. So it has a bunch of small proteins in it, around 300 or so, site saturation mutagenesis on each residue to every other. And yeah, then they performed that protease screening and then correlated that with delta G. And it lines up pretty well.
This one?
Yes.
Thank you.
Yeah.
So why is it delta G, not delta G? Because you're comparing a point mutation sequence with a native sequence.
Right. So when you typically think of stability, that's a delta G value. We're looking for the change in stability. So you're comparing two delta G values, which makes a delta G.
So I guess I missed it. The thing that's missing from protein MPNN by itself to make these thermostable proteins is that it's restricted, because what it learned is these proteins that both have to be stable but also do another function, and ThermoMPNN kind of releases that from this constraint, maybe? Or
Yeah, I don't think that's a terribly far-fetched explanation. Yeah, so what ProteinMPNN has been trained to do is recapitulate the native sequence, which has a lot of other pressures on it besides stability, including solubility, function, etc. And yeah, you can totally imagine that perhaps this adapter here that we've added on at the end is extracting the stability parts of that information that it's learned and amplifying that information, that signal, to better, make this prediction. Can't say exactly if that's what's happening, but yeah, that's I think a reasonable explanation.
Can it work on any type of protein?
Like
multimeric transmembrane protein.
So the data set were monomers, and these are only point mutations. I don't remember the numbers offhand, but I know Henry has looked at comparing the prediction at an interface, still a point mutation on one of the sides, comparing that to a data set that we got a little bit from the Baker lab, but I don't think the correlation was amazing. So yeah. Monomeric proteins. They're also, the proteins were limited in size, but it does seem to scale with some of the other data sets that have been created with larger proteins as well. There is one major limitation to this, and that's due to the assay itself, that it was the data that it was trained on. Predictions involving cysteine are often unreliable, and that's mainly because if you mutate something on the surface to a cysteine, it's easier to form a disulfide with another copy of itself, which makes it harder to cleave that protein. So it's often going to be more stabilizing than in reality. So often when we use this in practice, we often exclude predictions to cysteine. Okay.
What about prolines?
The prolines honestly weren't that much of a concern. It was mostly the assay's limitations for the cysteines forming disulfides. Okay. The second tool I wanted to talk about is AF2-Initial Guess. We've heard this term, this phrase quite a lot in the bootcamp. Amrita talked about it yesterday during her special talk. So I wanted to add a slide about kind of what it's doing and what the intention of it is. So it's still AlphaFold 2 in the sense that we take a design sequence and we're predicting a structure for it. In this case, we're specifically using it for binder design. So we have a target protein that is being bound by our binder. We have designed this, so we've maybe used RF diffusion to make the binder appear here. We've designed that sequence of ProteinMPNN. At this point, we want to see what AlphaFold kind of thinks of that interface, that confirmation that it's predicted. But what if, when we throw it through AlphaFold 2, it predicts it in the wrong site? That's not really useful. That doesn't tell us how much it likes this specific interface. So initial guess is kind of a way to make that happen. So what's essentially happening is we give it ahead of time the structure of the complex you're interested in, the complex that it was designed for. And then we use this as templating information or recycled information. So remember, AlphaFold has these 3 recycles. It makes a prediction. You give that prediction's information back to the model and tell it to try again. This case, we kind of give that recycling information ahead of time. We tell it, oh, you've already predicted it to look something like this. So maybe predict it again. And a lot of time it does listen to that and will repredict your structure. When you're doing that, there are some considerations. When you give it the full sequence of your binder, it knows exactly what, it matches exactly what's in the template, which means AlphaFold will most likely be overconfident in its prediction about how good that structure is because it matches exactly what's in the template. It was trained to, if you saw it in the template, to essentially recapitulate it because that's how you get the best training loss on the PDB. And a way to kind of avoid that is when you do this procedure, you typically mask the template sequence. So it says that we know the binder, the target sequence, we know where the binder is, but we don't know its sequence. And so what this will do will still kind of refold the target, but it still place it here, but also kind of give an interesting metric of its confidence and how it likes that interfacial site here, this epitope. And the way it does that is really the confidences. So if you look at the PLDT, the PAE, if this is a really bad sequence for this particular interface, those will probably be really bad. And it tends to like things that score, so things that score good tend to also behave better in experiments and things like that. So this has been at least validated a little bit in the Baker lab, and I think other people are also seeing success with it.
So the idea of giving it the first step or already the template there, it goes against the notion of we want it to bind to a specific place, because as you're saying, it could bind to other places, but if you're forcing it to bind to a specific place, it may fall into a local minima there, right?
Well, I mean,
so that works for, and I think I agree with you, that works if that's the only interface that it will make sense, right? But what if the binder wants to bind to another place as well, right?
I mean, it may want to. But the question that this, I think, this procedure was designed for was mainly asking, what does AlphaFold think about this designed epitope? So we're specifically designing the binder for this spot. And it's asking AlphaFold what it thinks of the design sequence there, the interactions that it's forming. Of course, you still want to run probably a multiple prediction without this, because if it does predict over here, maybe there is something, a concern. But this is really asking more of a specific sequence-based question, interaction-based, confirmation-based.
How often do you see them flying away, like not like losing fully the interaction between them?
It's not terribly often.
So doesn't that mean that it is now really overconfident about that interface?
Well, to kind of to change that, we mask the template sequence, and that really dramatically changes kind of what AlphaFold's seeing and what it thinks of this interface now. Because it's no longer certain that it saw exactly this binder there. It saw the shape there, but not this exact binder with that exact sequence.
So, I'm going to speak on behalf of the maintenance Chelsea because proteins are very much a case-by-case basis. But AlphaFold 2 multimer does horrifically bad at antibody and T-cell receptor docking. So T-cell receptors are a 3-body problem. And oftentimes what AlphaFold will do is it'll stick the TCR and the MHC, and then it'll pop the peptide that's supposed to be between the two of them off like 50 yards to the left. So there have been other methods that came up with using templates to fix that sort of docking, and things don't really seem to fly away when we do that, but basically what they do is try to put in all the different degrees of freedom for the template that we're interested in, and then they use confidence to dock what the best thing to do would be. So it's better to have like a controlled experiment in that sense. There shouldn't really be a situation where it's flying away given a template.
It doesn't happen often.
So this is only useful for like binders or like protein-protein interactions, not like small molecules.
Well, I mean, remember, AlphaFold 2 doesn't take into account the other molecules in the system. There are things that like try to fill in spots that may be, like harbor a good site. Like there's AlphaFill that will try to fill in specific molecules into regions that it thinks may be actually trying to coordinate it. But yeah, this was specifically designed for PPI's binder designs specifically.
Can you use LigandMPNN to generate the side chains, and based on the confidence that generating those side chains, assume something similar?
I don't, yeah. I mean, I think that's a reasonable assumption as well. It'd be interesting to see how that correlates with like this predictions, but I think that's a reasonable thing to try. Okay. The next one is AF2 rank. I think this is just a really convenient way of testing decoys and assessing their quality. So what Sergey and Roney found were that AlphaFold's confidences are really good at figuring out what works and what makes a good structure, what makes a good interaction. And so this kind of points to the fact that AlphaFold has kind of learned a nice energy function that works really well for proteins. And by using this specific score, this composite confidence score, they're able to really discriminate bad predictions, bad decoys, bad poses from things that are much, much closer to the native structure. So this composite score is made up of the PLDDT. You multiply that by the PTM, and then you compare the PTM. So this is literally the template matching score between the predicted structure and your original structure. Because if those match, that helps this term. And you want to make sure that these guys are confident as well. And this has been shown to be, I think, I don't think I've seen anything recently that has been better than this. So this, I think, is maybe the state of the art currently for decoy discrimination. And it's much, much better than Rosetta and some of these other methods.
For decoy discrimination in practice, would you know the ground truth structure to calculate the TM to it?
So what you're calculating the TM to is your decoy that you're giving it and the prediction of that decoy sequence. Yeah. All right. The last one I wanted to talk about was BindCraft. This is just a recent protein design method that is more or less automated. And it takes full advantage of AlphaFold 2 multimer's also learned energy function. It also uses soluble protein MPNN and then some filtering with AlphaFold and Rosetta and some other things. So the main idea is we have a target protein we want to design a binder towards. What we want to do is design a binder that has a nice interface. It contacts the protein very nicely. AlphaFold's also confident in its sequence and stuff like that. But remember, AlphaFold only operates on, you give it a sequence, it makes a prediction of that structure. So how do you get it to design its own sequence, what it thinks of a good sequence? So what we do here is backpropagation through the network. So we tell the model what we want to see. We want a binder with high confidence, a nice amount of interactions, low PAE, etc., maybe some other differentiable constraints. And we make a prediction. So we have random noise for the initial sequence. This is continuous noise. So we, so it's actually not a discrete sequence. It's 20-dimensional because it's L by 20 now. So there's a probability for each amino acid. We throw that through the model. It makes a prediction of what the backbone structure for that protein should be. But it's probably going to be bad at first because it's kind of random. So we start to compute a loss based on its predicted confidences for that structure. Based on that loss, we can backpropagate that to provide a gradient signal for how to change the sequence. And by changing the sequence, we can start to change them in such a way to maximize the confidences, to improve the number of contacts, and essentially improve what AlphaFold thinks a nice interface looks like. And so you can backpropagate, update this, and eventually you'll get to a place where AlphaFold is pretty confident and you're pretty happy with the scores that you're getting. And then at that point, they fix the sequence that's at that interface that AlphaFold designed and it really likes. And then they redesign the rest of it with soluble protein MPNN. So the rest of it, I don't know why particularly, maybe it's not as soluble or something like that, but just to be safe, you redesign the rest of it because that will be self-exposed if you've designed this properly. And that will allow it to be more soluble and ideally be better optimized for the design. After that, they do some validation and filtering, so they throw it through AlphaFold monomer, make sure it folds up into the same binder shape, make sure that it does contact this interface with a multimer prediction, and then you can do some filtering based on confidences and Rosetta scores and stuff like that. By performing this process, this workflow, they were able to design binders for a variety of different targets with very high success rates. So 13 out of 53 designs, 7 out of 9 designs, 3 out of 9, 4 out of 16, 6 out of 6, 4 out of 10, 4 out of 7, 2 out of 7, 6 out of 11, and 3 out of 3. So fairly high success rates. And I think a really cool thing is they got high success rates with a small number of designs, which is really, really important to see. And these are fairly tight binders, some nanomolar and even sub-nanomolar. And micromolar, so fairly good affinities as well. Yeah.
How does this compare with Bennett's design pipeline that uses AlphaFold 2 Initial Guess?
How does it compare in terms of success rates, like experimentally or?
Success rate, yeah, I guess what you would do.
Yeah, so this would require more compute to do because you have to spend iterations optimizing that sequence and optimizing the confidences of AlphaFold itself. And then, soluble MPNN's fast enough, and the filtering is probably fast enough. So I think this would require more computational resources by designing a sequence for that interface. However, on the other hand, for like the DL binder stuff, you would have to diffuse a backbone, design a sequence. So that cost, you're kind of trading off the cost between multiple AlphaFold 2 forward passes and backward passes for the computational cost of RF diffusion, which I mean, depending on how many backbones you have to design with RF diffusion, may or may not be equal. This may be slightly cheaper. I don't know. They're probably fairly similar.